Welcome everyone to my talk. As we just heard, it's about a novel group of giant viruses which we discovered in metagenomic data. And this particular group of giant viruses is pretty outstanding, as they encode for many more genes potentially involved in translation than any other virus known to date. And you might wonder what giant viruses are. So you can see here a lot of different giant viruses, and it's clear they can be quite divergent in their shape. But what they all have in common is that they have extremely large virions, which can range up to 1.5 micrometer in diameter, like we find it, for example, in pitovirus. And they also have extremely large genomes with up to 3 megabase. At the moment, the record is held by Pandora virus. And both particle size and genome size are fairly in a range of what is typical for cellular organisms. All giant viruses we know to date infect eukaryotic cells, in particular protists, and we can roughly divide the giant viruses in two groups. So the first one thrives in modulated vacuoles, so-called virion factories. So they're like in the cytoplasm of the host cell, while the other group makes use of the host nuclear machinery for the replication. And here you see a bit in more detail how such an infection looks like. So this shows cytoplasmic giant viruses. We see here a eukaryotic cell surrounded by virus particles, and they get taken up by phagocytosis. And once they're inside the host, they unload their genetic material and initiate transcription and also protein biosynthesis. For that, they make usually use of the host translation machinery. And here we see how those new virions build up in this so-called virion factory. This process, what I just showed, is only partially understood. So we really lack detailed knowledge about molecular mechanisms underlying the course of infection. And when we look in the genomes of giant viruses, we see that what we currently know is actually very little. So the function of most of the genes encoded in giant viruses is currently still unknown. While we also find some genes with homology to their cellular counterparts. And that brings me to one of the most intriguing findings upon discovery of giant viruses around 15 years ago, where they discovered a couple of genes potentially involved in translation. And that kind of is against the central dogma that viruses are not live. So one of the hallmarks there is that viruses do not perform translation. So it was kind of puzzling to see genes potentially involved in translation in these genomes. So among those genes, they found aminoacyltRNA synthetases, translation factors, tRNA modifying enzymes, and also tRNAs. And this finding led to, or gave rise to two different hypotheses of how giant viruses might have evolved. So we see here on the left the first scenario in which we start with a rather small virus which infects its eukaryotic host, and then keeps taking up genetic material from the host through horizontal gene transfer. And this genetic material can also come from digested food, what the host takes up in addition. And over long periods of time, this process increases the size of the viral genome, and we end up with a giant virus. While the other scenario, what we see here, starts from a cellular organism, which is either like an ancient eukaryotic cell or even a member of a elusive fourth domain of life, which eventually is already extinct. And giant viruses evolved from this kind of cell due to some trigger where the cell learned to become a virus. And then through constant reduction of the cellular genome, we end up with what we observe in giant viruses. In both scenarios, however, there should be a virus which has a genetic complement much closer to eukaryotic or to cellular life, which in the first scenario would be a much more highly derived virus. So it took up many more genes from the host. While in the other scenario, this should be kind of the snapshot of the transition of such cellular organism to virus, where the genome is not yet that reduced. And finding this kind of hypothetical, more highly derived virus would provide the ultimate chance to study those additional cellular genes and to test for any of these two hypotheses. And such a virus could provide the missing link to understand giant virus evolution. And it seems that we have now found this missing link. So in the course of ETOP with the University of Vienna, we performed metagenomic binning of Raman-sorted nitrifying microcolonies, which come from a wastewater treatment plant in close to Neuburg, Austria. And in the course of this work, we detected a clearly separated metagenomic bin. So this shows a coverage versus GC plot, and we see the contigs. And in green, these are contigs which could be identified as being of viral origin. And this is just one example. So we screened more than 100 mini-metagenomes, and we find this virus in at least 12 of them. And in addition, we find this virus also in deeply sequenced metagenomes from the same sampling site and in metatranscriptomes, like we find the genes expressed. So making use of this data, we could then assemble a genome of a potential giant virus with a size of 1.6 megabase. And it shows many typical features of giant viruses. As we see here, like in the outermost track of this plot, all genes highlighted with blue circle, which are uniquely shared with viruses. And we see genes highlighted here with red circles, which are uniquely shared with eukaryotes and not found in other viruses. And also when we look in the center of this plot, we see all these connecting lines. So this visualizes many paralogs or duplicated genes, as this is one of the key features of the giant viruses, that they massively expand the gene families. And that's also the case here for this virus. And based on its origin, we termed the virus Klosner-virus. We then took all the genes of Klosner-virus and placed them in a gene-sharing network, together with a representative data set of archaeal, bacterial, and eukaryotic genomes, and all the available giant virus genomes. And we see that Klosner-virus positions itself centrally in between viruses and cellular life. So it was pretty exciting. And we tried to find additional related genomes, so like additional viruses. For that, we took the all Klosner-virus genes and used them as a bait to screen through around 7,000 metagenomes available in IMG. And we could detect at least 3 more closely related potential giant viruses. One comes from a wastewater treatment plant in Cape Town. We denote it as catovirus, and it has a similar genome size as Klosner-virus. And another virus came from a wastewater treatment plant in Hong Kong. We call it Hokko-virus, and slightly smaller genome and a bit lower GC content, actually the lowest GC content of any giant virus known to date. And then we have a comparably smaller giant virus, Indy-virus, which comes from a lake sediment from a lake in Indiana. And altogether, we denote this novel group of giant viruses as Klosner-viruses, and we then place them in a phylogenetic tree together with other giant virus lineages, and we see that they position that they're in a sister position to the mimiviruses, and it's also clear that the close evolutionary relationship to mimiviruses when we look at the gene content. So we see that across all the different combinations of giant virus lineages, most genes shared between lineages is between mimiviruses and Klosner-viruses, which is just around 120 gene families. And that's a comparatively low number when we think of about 2,500 gene families which we add to the known viral coding potential by what we can find in these Klosner-virus genomes. We then look for conserved features. So these are usually genes which are needed for infection process of giant viruses, that they really function as they should. And we see those genes plotted here, like presence and absence, and also copy number. And we see that closnoviruses also here resemble very well what we find in mimivirus genomes. But there are like a few differences. For example, we see several additional copies of capsid proteins and also of this gene called packaging ATPase. So both potentially contributing for like a higher versatility in host interaction. And next we try to model the most likely path of the evolution of the closnoviruses. So for that we go along the species tree, and we see here, like in blue, events of gene gain. In red we see events of gene loss. And we also see gene family expansion and contraction. So all these are mechanisms which usually like shape these giant virus genomes. And here we start with a rather small virus, around 240 gene families. And we see that through lineage-independent gene gain, all these different lineages here emerge. This is kind of a highly dynamic process, as it becomes clear when we compare closnovirus and indivirus. So, as I said already before, indivirus has a much smaller genome, and we see that in contrast to closnovirus, which had here like a massive gene gain, indivirus was subject to more subject to gene loss, and also not to that extent to gene family expansion, but rather to gene family contraction. The most exciting finding from these closnoviruses is that they encode many more genes potentially involved in translation. So I mentioned this earlier. And when they first discovered giant viruses around 15 years ago, like the mimiviruses, they found up to 7 amino acyl tRNA synthetases. And in closnovirus, we already see 19 different amino acyl tRNA synthetases. And taking the closnoviruses as a whole, we find these genes covering all 20 different amino acids. In addition, we have also several more transcription factors and tRNA modifying enzymes. And thus, when we think back of these models of giant virus evolution, it seems that closnovirus could provide this missing link to study which of these two scenarios might be the correct one. And we did that, so we took all the genes potentially involved in translation and performed like a large-scale phylogenomic analysis using those genes. And here are the potential outcomes, like simplified. We see the tree on top. This tree would support reductive evolution from like a cellular ancestor. In that scenario, the giant viruses should branch at the root of any of the cellular domains, while the lower tree shows the outcome in case of uptake of host genes through horizontal gene transfer. Here we would find giant virus genes within cellular domains and potentially also being polyphyletic. So that means that different giant viruses have acquired this certain gene from different hosts. So we performed this analysis for, as I said, more than 40 genes. And here's just a summary of how the trees looked like. In most of the trees, the closnovirus genes branched within the eukaryotes, and they were also often polyphyletic. There are only a few cases where they were positioned at the root of the eukaryotes, and this often can be attributed to tree-building artifacts. As we handle here very divergent sequences, we have to keep in mind. And altogether, this clearly gives evidence for the scenario of uptake of host genes through horizontal gene transfer. And looking at these two scenarios, we now definitely can refute the hypothesis of the evolution of giant viruses from a cellular ancestor. With that, I'd like to briefly sum up the findings. So we discovered a new group of giant viruses, the closnoviruses, and this was done completely independent of the host, so from metagenomic data. And they currently represent the largest genomes of cytoplasmic viruses. We had more than 2,000 novel viral gene families to known viral coding potential. And they encode for this highly expanded viral translation system, if you want to call it like that. And provide this missing link, what I mentioned before. And after studying all these highly interesting genes, we can now clearly say that our finding gives enough support to say that giant viruses definitely evolved from smaller viruses by evolution, by horizontal gene transfer, like acquisition of these genes from their host cells. With that, I would like to thank people involved in this project. That's our internal support from JGI and our collaborators from the University of Vienna and also from NCBI. With that, I'm at the end and end here, and I'm open to take any questions.
Yeah, I have two very different questions here. I guess I'll go to what I consider the minor one here. Given all the parallel translational machinery, do you see, and there's that class of them that are cytoplasmic, do you see them as a potential good molecular factory for whatever products, especially if we can put in alternate tRNA synthetases and the like? And the other question that I consider more major here, I read part of a paper a long time ago that tried to hypothesize that the origin of the eukaryotic nucleus was a virus particle, which theoretically, in line with this, you could have a virus coming in, helping to form the nucleus, and then popping out periodically. Could a hypothesis like that be supported by your data?
Yeah, so I think, thanks for the interesting questions. I'd like to start with the second question. As this also, it was one of our initial motivations, as I was really excited about this hypothesis that nucleus evolution, like in the eukaryotic cell, and giant viruses, that there might be a connection. And so that was one of the initial things we want to see. And so in that scenario, we would also expect that these genes follow like a similar evolutionary history. And based on our findings, so this is definitely not the case. So we can exclude that there was this connection between eukaryotic nucleus and the virus. And we did not follow it up further then. So it didn't fit anymore, like based on the phylogenies, like what we see in the trees. And the first question, I think I did not completely get it. So you asked for if we see viral factories or if we would expect, or... Okay, so it's a good point, but I think nobody really has tried this so far. It sounds definitely interesting. I mean, we don't know what the closenoviruses do. We've not really observed them in vivo, so it's just metagenomic data. So based on their genetic complement, we think they're definitely cytoplasmic viruses, as they're so similar to meme viruses. So they would build up these viral factories, and the exact mechanisms happening in there, in regards to the much more comprehensive translation system, we don't know how this influences the actual course of infection, and if these genes are actually being used, which would be exciting. And having like a model like in the lab where we could study this would be great. Like, we are not there yet, but I hope soon. And so at the moment, I have to say, I don't know about the possibility to set up viral factories in other cells. Yeah.
Yeah. So thanks for the great talk. So we seems like to go into this hypothesis and finally having some answer about like where the giant viruses come from, and this is this kind of increase in genome size with time, which causes another problem, which is usually capsid of a virus is tightly linked to the genome size, and so. Do you have any clue or any guess or anything from these new viruses that will suggest how they kind of increase their capsid size with time as well?
Mm-hmm. Well, it's also very interesting. I thought about this earlier, and when you think back of the first couple of slides I showed, so we see that like pithevirus, which has kind of the largest particle size, has actually a much smaller genome than Pandora virus, which has a smaller genome size. But I think both picovirus and Pandora viruses don't really have capsids. So we have to step down to mimivirus. And so we know mimivirus can harbor like 1.2 megabase genome. And we have now additional copies of capsid. We don't know what is the maximum of genome what we can pack in there, or if there are certain strategies to better fold or compress the DNA. It's imaginable. So we know from bacteria that they're very, very tiny bacteria with rather large genomes as well. But I mean, you're right with the capsid, the way it's built. So it's puzzling. But still, I feel like what we see in closenoviruses with 1.6 megabase genome, that should be doable, I guess. If we already know from mimivirus that 1.2 megabase fit in, that's not that much larger, maybe. Yes, it's hard to tell.
Is there any progress in knowing what the host organisms are?
Yeah, also a very good question. So, as I said, we did not isolate these particular giant viruses in contrast to all the other giant viruses known to date. I think they all have been isolated in co-cultivation with Acanthamoeba. There might be a few exceptions where they found a giant virus like in the marine ciliates or so. But concerning the host for closenoviruses, so we checked, we assembled 18 S sequences in these metagenomes, and we also looked what is expressed in the metatranscriptome data. And we, for example, didn't find Acanthamoeba in there, which is at the moment, as I said, the common host for giant viruses. So we would assume that this is not the host, Acanthamoeba. We found some consensus of protists which appear in all in all 4 metagenomes. And that is like a protist phylum called Cercozoa, but it is not so helpful to us now. We could speculate about it, that this is the host. Like Cercozoa are pretty diverse. And also when we think of the genetic complement of these 4 closenoviruses, also they only share like a small fraction of their genes. And there was like a hypothesis that giant virus genes get really shaped a lot by the hosts in which they thrive. So we would expect based on this that they might not share the same host, but rather different protist hosts. So at the moment, I cannot really clearly answer the question, what is the host of closenoviruses? We have some leads, but first they need to be isolated together with the host, and we know.
Okay, last question.
Hi, Frederik, we're here. So thank you for the talk. So I have a curiosity as well. So since you have the metagenomes, right? So have you tried to look for virophages in those metagenomes that you can somehow try to correlate with this giant virus as well?
Sure. So that was also one of the first things we did, because we know that these giant viruses, or like let's say the population might get affected by phages, so viruses which infect viruses more or less, and that's pretty cool. And so yeah, we had a closer look, and at least from the Kloster-Neuburg sampling site, in none of these metagenomes we found virophages. But I think in one of the metagenomes where we found, I think it was indivirus, there we also see virophages in the same sample, but that doesn't mean that this virophage is in particular focusing on indivirus. And at the moment, there was not a really clear connection. We would expect it, I think, for mimiviruses, they find virophages for all of them, and also for cafeteria virus, which is also part of this called extended mimiviridae, there are virophages. Closenoviruses, we don't have them now at the moment.
Okay, let's thank Frederik again.